So, I mean, I guess my talk will be mostly about a little bit different from, I will talk about narcolepsy, autoimmunity. So I think we're really at, I mean, I'm fairly happy because I think we are very close to finishing up pretty much what we need to know about narcolepsy. I think we made a very major discovery, which is going to be published in PNS, I think this week or next week. Which is going to really help, I think, to close narcolepsy autoimmunity. And that's what I would present today. So you probably know about narcolepsy that it always has been maybe one of the way. I mean, we know that sleep is very complex to maybe understand sleep and sleep regulation. And it was first described in the 19th century. And then in 1918, there was this very weird kind of flu that came around. That was the Spanish flu. It killed about 100 million people. And then a lot of people had this residual symptom, or at least we don't know. It was kind of a separate epidemic that started a little bit later, where some people would just go into some kind of coma and be sleeping all the time. And then if they recovered, they had Parkinson's disease very often. And it was called von Economo encephalitis lethargica. And this guy, von Economo, really sliced the brain of these people and tried to figure out, I mean, we know that there were brain damage. Half of those people died, so it was not just a very, it was very severe disease. And when you look at the brain, they had cell infiltration in different brain areas. And then when you looked at the brain of people who were sleeping all the time, this encephalitis lethargica, there were a lot of lesions in the posterior hypothalamus, which contained the hypocretin or orexin neurons and the histamine neurons. So in fact, it was probably the first to kind of suggest that the posterior hypothalamus was very important to regulate wakefulness. And it's very interesting that, in fact, it was also connected to the flu, because you will see that at the end, narcolepsy is really connected to the flu. I must say that it's probably a different disease from narcolepsy, because these people didn't really have clearly cataplexy. They were just more in some kind of coma. Then in the 30s, the first stimulant treatments were developed, like with benzedrine, which is basically amphetamine, which is a modification of norepinephrine or dopamine, which is just methylated so that it goes more into the brain. And then it kind of catecholamine that's modified. And then in 59, it was first discovered that patients with narcolepsy go straight into REM sleep. So the idea is that narcolepsy was maybe a REM sleep condition came about. And indeed, there are a lot of symptoms of narcolepsy where patients are either paralyzed while awake. So there is this sleep state dissociation where patients are half into awake and half into REM sleep. And that explains a lot of the symptoms of narcolepsy. They go very quickly into REM sleep. Sometimes they when they laugh, and that we don't really understand fully, they kind of get paralyzed the same way as you are during REM sleep. And then very often they have hallucinations where they dream while they're still awake. So there are a lot of this sleep-wake dissociation that corresponds to the fact that they have very abnormal sleep, but in particular they can't stay awake and they go very quickly into REM sleep. In the 70s, there were dog models that was developed. And then in 1983, there was a complete surprise because no one really knew much about narcolepsy. I mean, it was believed to be really a key to maybe understanding sleep, but it was considered more a neurological disorder, a brain disease. And then suddenly, when some people started to look at this series of genes that are called HLA, that are very important for the immune system, and they discovered that almost all patients with narcolepsy had a specific HLA subtype that was called DR2. That was discovered in 1983. And then later on, we found that it was actually not DR2, but another gene called DQB10602 that were involved. And, but still that it had something to do with the immune system. So starting in the 80s, people thought narcolepsy must have an immune component, but nobody could really find what was the target of this immune or autoimmune component. And then in 2000, we had this dog narcolepsy that we clone that had a mutation in the receptor for hypocretin or orexin, and that kind of raised the possibilities that it was really this orexin system was very important to regulate wakefulness. And then now I think we understand that in humans, it's not the receptors that's broken, but actually the hypocretin neurons that are missing, and it's because of an autoimmune process. And as I will explain, what's really amazing is it's coming back full circle to von Economo in some ways, because it seems to be involving the influenza as really the trigger for the autoimmune process that destroy hypocretin cells. So really the genetic epidemiology, as I said from the beginning, was consistent with an autoimmune disease. So for some of you that don't know much about autoimmune diseases are very common. And there's about 5% of the population that has autoimmune disease. There's rheumatoid arthritis, where it attacks your joint. There is multiple sclerosis, where it attacks your myelin sheaths and you have problem of nerve conduction and you're weak, or you have problem with vision. There is, autoimmune disease affect pretty much every organ. But very curiously, there are very few autoimmune diseases that affect the brain. Even multiple sclerosis is actually mostly disease of the brain that affect white matter, so more like the oligodendrocytes. So curiously, for a long time, people thought that the brain was immune privileged, but it's actually changing quite a bit now. People are really believing that there are probably a lot more autoimmune disease or immune disease of the brain, but that we have trouble detecting it because the brain is so plastic that maybe the symptoms are not very clearly defined. To give you an example, even in Parkinson's disease, when you do a genetic screen of Parkinson's disease, one of the top genes you find is actually HLA, which is again immune related. When you do schizophrenia, the top genes that you find is HLA. So again, there is probably a lot more autoimmune disease of the brain or maybe disease that involve infections, because autoimmunity and infections are very linked, that probably have to do something to do with the immune system. So it's a very interesting area. And just to explain a little bit for narcolepsy, we have this very, very tight association with HLA, which initially was discovered in 1983. But then when we did more fine mapping of exactly which HLA genes was involved, we discovered that it's not DR2, but an other gene called DQB10602 that's very close to DR that's involved. And it's really remarkable because you absolutely must have this particular variant of HLA to develop narcolepsy. Again, 98% of patients have it versus about 25% of the general population. So it's almost necessary, but it's not sufficient to develop narcolepsy. And then we know that narcolepsy is not just genetic in humans, not like dogs. You also have some environmental effects that must trigger narcolepsy, because you take twins, they're exactly identical, and then usually only one has narcolepsy, not the other. So something must trigger narcolepsy in one twin and not the other. But there's still some other genes that are involved, because if you just looked at family, if you have narcolepsy, there's still a lot of increased risk for one of your family member to develop narcolepsy. It's about 1 or 2%, 1% for cataplexy, which is much more than the general disease prevalence, which is 0.05%. So if you have narcolepsy, there is about a 30 times more chance for one of your children to have narcolepsy, even so the risk is only 1%. It's still low, but it still shows that there's probably other genes that are involved in triggering narcolepsy. And all this kind of autoimmune disease, I could have exactly the same slide for type 1 diabetes, for example, which is an autoimmune disease where you destroy your insulin-producing cells. It's exactly the same. There's a high association with HLA, environmentally influenced, increased familial risk, and we know it's an autoimmune disease, or multiple sclerosis, etc. So there's a lot of this autoimmune disease, and most of them are associated with HLA. And we really don't understand completely how it works. But the idea is that these HLA genes, there's actually more than one. There's a whole series. They are all on chromosome 6. But what's very remarkable with these HLA genes is, actually I have a fascination for HLA, is that they are very polymorphic. So most genes in your body, they are the same for everyone, because of course if you change an amino acid in one of your genes, it's not going to work as well because it has a different structure, and then it's bad. It's going to be a mutation. But actually for HLA, it's the opposite. It's one of the most polymorphic genes in the body, so that these different HLA genes have many, many variants. And in fact, they were first recognized as very important for transplantation, because from one person to the next, you always have different variants of these genes. And that explains why it's very important to match for transplantation. But of course, transplantation is not the normal function. We didn't go through natural selection based on transplantation. And the real role of this HLA molecules, which was discovered actually quite a while, is to explain the genetic diversity of the immune system. In fact, most of the genes that you have in your body, they are either regulate the brain or the immune system. I mean, permanently we are confronted with all kind of immune, our guts, are full of microbiome, of cells, and we have always to fight, these cells and keep them in check. As soon as you die, you first rot from inside, and not to be poetic, but it's definitely sure. We are really confronted to all this virus and bacteria permanently, and the immune system is really important to keep that in check. The problem is at the same time, it needs to be sure that it recognizes its own self, that it doesn't destroy its own self, and that results in autoimmunity. And the way this recognition of the foreign peptides is through this HLA system. And the way it works is the HLA is very polymorphic. So this is, for example, the HLA DQ that's associated with narcolepsy, and all the different amino acids that differentiate different people across the different HLA subtypes that we have, they are all in this binding groove here, and it binds fragments of virus and bacteria, which are very, then are recognized by a series of cells that are called T cells, that then develop an immune response against pieces of virus or bacteria. So you can imagine that if you have a different HLA subtype, let's say that I have DR1 and someone is DR2, your DR1 may bind a certain piece of influenza, a certain piece of the virus, and DR2 another piece of the virus, and then it's going to be recognized by T cells differently. So of course, having different HLA type makes you have a different immune response to the same virus. And in fact, it explains why there is genetic diversity of the immune response, which is very important because that allows the population to be better protected against infection. This way, if tomorrow we have a new flu that arrives, some people are going to be HLA DR2 and bind a certain piece of that flu, other people will bind another piece of the flu, and this way, we are going to be better protected and have more diverse immune response, so that this way the virus cannot mutate just one piece and then everybody would die because it wouldn't be recognized by the immune system. So really, these HLA are very important to explain the genetic diversity of the immune response. And HLA genes are associated with infectious disease. For example, if you have a chronic hepatitis, you have certain HLA genes. If you have lupus, of course, depending of the HLA genes, your immune response to infection is going to be different. But the diseases that are the most tightly associated with HLA, like very, very strongly, are usually autoimmune disease. And the hypothesis is that the problem is sometimes you have this particular HLA type, and instead of just binding a piece of virus, sometimes a piece of, for example, or yourself, in this particular example, is going to be hypocretin itself, or orexin, is being confused with a piece of the virus, and then it being recognized similarly by the T cells, and instead of the T cells only destroying the virus, it starts to destroy your own self and you develop an autoimmune disease. This idea is called molecular mimicry. There will be mimicry between an infectious agent that's being confused by yourself, and then you develop narcolepsy or an autoimmune disease. So this general idea of molecular mimicry would explain why there is this very tight association with HLA. But curiously, this has been demonstrated in animals. You can produce this kind of abnormal immune response, but in humans, nobody has really been able to demonstrate molecular mimicry. Nobody really has been able to associate autoimmune diseases with specific infection. There is some hints, but exactly how it works, this has never been proven. And interestingly, I think narcolepsy might be the first disease where we really can establish this, as you will see. So the first thing that we discovered a few years ago is after the mutation, we discovered that the mutation in the dog narcolepsy was a receptor for this particular peptide called orexin and hypocretin. But in humans, of course, as I mentioned, it's much more complicated. So clearly, it was not a mutation. It was actually the lack of hypocretin neurons that caused the narcolepsy. And indeed, when you look post-mortem in the brain of a narcoleptic patient, this is a normal person, and you see all the hypocretin neurons. And then here is a patient with narcolepsy post-mortem. You see all the hypocretin neurons are gone, and we can actually measure the levels of hypocretin orexin in the CSF. And in patients with narcolepsy, that of course has this HLA DQB10602 type that I told you is very important for the immune response, it's generally all the hypocretin neurons are gone and you have no more hypocretin cells, and that's why you have narcolepsy. So the good news is actually that right now, patients, there is actually several companies that are trying to create hypocretin agonist or orexin agonist. In fact, there's 2 companies that have succeeded and that are now starting clinical trials. So of course, that's going to be the ideal for narcoleptic patients because you are going to replace what's really missing and what's producing all their symptoms. So that's why when I'm telling you that I think narcolepsy is kind of half solved, is, once we have the hypocretin agonist or the orexin agonist, I mean, for sure it's going to be a very good treatment for patients with narcolepsy because that's what they are lacking. A little bit like insulin, type 1 diabetes are lacking insulin, and insulin is, of course, the best treatment. But, of course, the next question is exactly, why these hypocretin neurons are destroyed and how it happens. And as I mentioned, because of this HLA association, has been suspected for a long time that it's an autoimmune disease that attacks a part of the brain, and then, of course, hypocretin neurons will be the target. The problem is, after that, it became very complicated. We discovered this link with hypocretin in 2000, and since 2000, I've been struggling in the dark to try to figure out exactly how it happens. One of the reasons is that in most autoimmune disease, you find antibodies against the target. Like if you do one classical autoimmune disease that looks very much like narcolepsy is type 1 diabetes. It's a disease where you attack your neurons that produce your cell is the pancreas that produce insulin, and then you destroy your insulin cells, and then you develop type 1 diabetes, and you need this injection of insulin for the rest of your life. This is very similar in narcolepsy, we believe, with the lack of orexin or hypocretin neurons, exactly the same, except it's the brain instead of the pancreas islets. However, in type 1 diabetes, you find a lot of antibodies against the insulin-producing cells. So it's very easy. You can take serum of patients with type 1 diabetes, and then you slice pancreas, even in rats or in humans, and then you put the serum of patients or the serum of control, and the serum of patients has a lot of antibodies that are going to recognize the insulin-producing cells, surface antigen, insulin itself, and then as a consequence, you get really this labeling of the insulin-producing cells. So it's very easy to show that there's an autoimmune process against insulin-producing cells in type 1 diabetes because in the serum you have all these antibodies that recognize insulin or insulin-producing cells. The problem is that you don't really know what is secondary to the disease and what's really killing the insulin-producing cells. That is the main problem with autoimmune disease is what is the causal, destruction? Because once you get inflammation and then insulin-producing cells get destroyed, you have a lot of secondary immune response, and then it's very difficult to know, what is killing the neurons versus what's a secondary reaction to the destruction of insulin-producing cells. Because you see so many autoantibodies and T cells that infiltrate the islet cells. So for narcolepsy, unfortunately, we don't find any autoantibodies. We just try to slice the brains. Many people have tried, in the area where hypocretin neurons are, and then you add the serum of patients with narcolepsy, and they don't label the hypocretin neurons. So there is no antibodies that we can recognize. And we tried really hard. We even isolated the hypocretin neurons and found each of the main proteins that are overexpressed in these neurons and tried to see antibodies against orexin, hypocretin. We found nothing. So at the end, we got stuck. We said, my God, probably there is no involvement of B cells or antibodies, in the cause of narcolepsy. And so we switched back to genetics, and then we did more genetics, and then we did what's called a GWAS, which is basically you just screen all the chromosome for millions of SNPs of genetic variation, and you see what is associated with narcolepsy. And that gives you a survey of the genetic factors that explain why narcoleptic patients are at increased risk versus controls. And of course, when you see that, the first thing you see is HLA. I mean, I told you there's 100% or 98% have HLA-DQB10602, this variant that seems to be changing the response to a lot of infectious agent. And you have the p-value is infinite. I mean, it's 25% versus 98%. It's a huge p-value. And then after that, the other effects are very small. But still, there is one that's very strong after that, which is in the Tcell receptor. And that's almost a no-brainer. You may ask, would ask me, why did you even do the experiment? Because the HLA, when it binds a peptide, actually is recognized by the T-cell receptor. So you say, my God, I mean, that's obvious that it should have been the same pathway. But actually, it's a surprise, because in all of the autoimmune disease, you find very similar genes. That this one, Langerhans, is associated with type 1 diabetes. This one is associated with Crohn's disease. This one is associated with multiple sclerosis. This is associated with type 1 diabetes. I mean, all these genes have been shown to be associated with other autoimmune disease, but the T-cell receptor has never been shown to be associated with any other autoimmune disease, which really doesn't make sense because, again, I told you, HLA binds a peptide and it's recognized by the T-cell receptor. So it should be involved, but I will explain why it's probably not involved in other autoimmune disease. And what's really striking is both the alpha chain and the beta chain of the T-cell receptor are involved in associated with narcolepsy. And then another gene that's involved that we don't find in other autoimmune disease is this interferon receptor. It's very strong association. I will tell you a little bit more about that. So how does the immune system works? So the immune system has several types of cells, and I would say like the three main types of cells are the B cells, would produce antibodies, and then there is what's called the CD4 cells, which I like to call the general of the immune system. They are like the most important ones because they coordinate both the production of antibody by B cells, and they also coordinate with the destruction of virally infected T cells through another group of T cells that are called CD8 cells or cytotoxic cells. So basically, those T cells are just like the first line that kind of helps to recognize self and non-self and virus, and then coordinate the production of antibodies or the destruction of the infected cells by specialized cells that are called CD8 cells. So often we say that there is two different arms of the immune system. There is an arm that's an humoral arm, which is the antibody production, which I told you we don't find any evidence in narcolepsy. And then there is another one that's called the cytotoxic arm, with the CD8 cells that really kills directly the cells that are infected or so forth. And from what I told you, we don't find these B cells, but it really looks like the cells that are involved from the genetics is really these two groups of cells. As you see, the genes that are really involved are mostly here. For the we know that for the CD4 cells, they are recognized by HLA class 2, like DQ. So, for example, the DQB10602 is clearly associated with CD4 cells. So this seems to be the key interaction that is involved in narcolepsy. But then, as I said, it probably doesn't go through B cells, but probably through CD8 cells, because we also find other genes like perforin is one of the genes that's expressed in cytotoxic cells, and then it's being released, and then it produces holes in the virally infected cells and kills it. So our general hypothesis, which is really suggested by this autoimmune scan, this GWAS, is that you have the CD4 interaction with antigen-presenting cells, and then you have the CD4 cells that then activate CD8 cells that kill hypocretin neurons instead of virally infected cells. And you will see it's turning out to be correct. So one thing that's a little bit complicated, and I'm really sorry, this is the most complicated slide of the entire talk, is that actually the diversity of the immune system in the T cells, like the T cell receptor, I told you already HLA, you have different subtypes across different people. In fact, it's amazing. Like even DQ has like 25 alleles. So that's why even at the level of DQ, all of us have different HLA subtypes. It's extremely complicated. Yes. So, but what's really interesting is actually the diversity in the immune response is really created by this reshuffling of genes between there's little segments of mini genes, the TCR alpha and beta, that actually recombine together to produce this T cell receptor that then can be random. And then only the ones that recognize your own self, they are being deleted by the thymus to avoid autoimmunity. And the ones that are useful, they are going to be released in the general population. And then once you meet a certain piece of virus or bacteria, that's being recognized by HLA is presenting a piece of the virus of bacteria, the particular TCR is going to grow, and then your T cell grows. So most of the genetic diversity in the immune response in the T cell receptor is actually not genetically mediated. It's mostly mediated by, the diversity in this recombination. Then even in identical twins, the T cell receptors are quite different. If you sequence the T cell receptor of two identical twins, there's almost as much difference as between two unrelated people. So what does this genetic association then with TCR means? So we're quite lucky because what we found is that there's two genetic effects. One is that it changes an amino acid in the J24 area, and then another one, it just changes the expression of a very specific segment in the V region that's called V4.2. So our hypothesis is that in the response to the autoimmune process, there must be an involvement of this very specific subclass of T cell receptor that involve one particular segment, that's J24, and then probably one particular G beta that's 4.2. And because it's genetically associated, it may be causal. So the next question is, what are the environmental triggers? And more we kind of started to suspect that the flu was involved because, as you see, the onset of narcolepsy seems to be seasonal. So that everyone that starts narcolepsy, usually it's around the spring or the summer. And so it looks like people get infected during the during the winter, and then it takes 6 months, and then they destroy their hypocretin cells, and then they develop narcolepsy. So our general hypothesis right now is that there is a flu infection during the winter, and then you start an immune response against the flu, and finally you destroy your hypocretin neurons by mistake through this T cell interaction with T cell receptor and HLA that then destroy hypocretin neurons. And then one clue that we got is in 2009, there is a new flu that appeared. That was the swine flu. I don't know if you remember, but that was very dramatic because a lot of people got scared that millions of people would die because occasionally new flus appear and switch species. And that's what happened in 1918 when I told you the encephalitis lethargica that killed 100 million people. So when this happened in 2009, people really panicked. And in fact, they created some new vaccine, and I will tell you a little bit about that. But what's very striking is even in China, for example, we found a lot more cases of narcolepsy were triggered by this new flu. So we started to suspect that the trigger is the flu that starts the immune response that then becomes abnormal and cross-react with hypocretin neurons. And again, the flu, it's very fascinating. I mean, we'll never get rid of the flu. You have to be very admirative of the flu because it exists in every species. And for example, it exists in swine and in birds, etc. And sometimes humans get infected by two flus, like a swine flu and a human flu at the same time. And then the combination of chromosomes recombines, and then it creates a new mixed flu that once every decade or 20 years or 30 years jumps species. Then it can actually infect not only a human, but infect from one human to the next. And that's what happened in 1918, in 1957, and 1968, and then in 2009 with this swine flu. You basically had a new flu that kind of was a mix of a swine and a human flu that started. And when this happened, people are really panicked because, again, in 1918, 100 million people died. I mean, so they really created this new flu vaccine that was used only in Northern Europe, only directed against this flu. And what was very odd is the number of people developed narcolepsy after this flu vaccine. It increased the risk about 10 times. So as a consequence, it really strengthened the idea that the flu vaccine was really involved that maybe the flu was involved in the cause of narcolepsy. So the next question is, what are the T cells that are involved? And what we did is we screened all the flu proteins of the vaccine for those that bind O602. So we kind of found all the pieces of flu that bind DQB10602, and we tried to analyze the T cells that respond to it. And we also screened the hypocretin itself, the orexin itself, because that was the best target for an autoimmune process. And we found some, as you see here, that binds hypocretin and others that don't bind that bind DQB10602 and others that don't. And then after we screened all T cells, we kind of added these pieces of hypocretin and these pieces of flu to see which T cells are actually recognizing this combination. So it's like a very tedious issue. So what you do is you kind of, here is, for example, an example of people, how these cells here that we select by fax, they're basically a group of cells that can recognize this particular piece of virus in the context of DQB10602. So you have DQB10602, then the piece of the virus, and then it's labeled, and then we can recognize the cells that recognize exactly this piece of the virus, the T cells, and then isolate them and sequence them. And we screened then, I mean, it was a huge amount of work done by Dr. Guo, and we screened all the pieces of flu, and then we also screened hypocretin and others. And as you see is, of course, when you screen pieces of flu, you have a lot of reaction. A lot of T cells react because, of course, it reflects an external agent. But when you screen hypocretin or your own self-antigen, you really get very few reaction because it's tolerance. You know, these cells have normally been eliminated by the thymus. But one of the surprises is actually when we use the hypocretin that was amidated, which normally hypocretin is amidated before it's released, actually it reacted a lot. The immune system seems to have a lot of T cells that recognize this post-translationally modified version of hypocretin. And then we noticed that we had a lot more reaction in the patient with narcolepsy, both to a particular piece of flu that's called hemagglutinin 271 to 287, and also to hypocretin. So it seems to suggest that maybe there were an overreaction to that particular piece of hypocretin and also that particular piece of the flu. And we noticed that there were also an homology between the different piece of hypocretin and the piece of flu. So we are starting to see that it's actually a cross-reactivity between the flu and the hypocretin with one particular piece. So we then use the tetramer to isolate the T cells that recognize this particular piece of the flu on DQB10602 and the hypocretin on DQB10602. And then we discovered that there is a number of receptors that are actually common. For example, this V4.2 is found in both the hypocretin reactive cells and the flu PHA response, hemagglutinin response. And then we found some other piece of T cell receptor that involve a particular segment called J24 in a lot of hypocretin reactive cells. And when we did a network of all these different heterodimer of T cells, what we found was very striking, because what we found is almost all the piece of receptors that recognize hypocretin use this particular TCR segment that's called J24, and that's the one I told you is involved in the genetic association with narcolepsy. And this one, 4.2, we recognize that a lot of T cells that recognize both hypocretin and the piece of the flu actually use this 4.2, a particular 4.2 sequence. So now we are pretty sure, because the genetics is also associated with this particular segment. So now we are pretty sure that the cause of narcolepsy really involves an abnormal recognition of hypocretin through this T cell receptor when it's presented by DQB10602, that's called J24, and then also this piece of hypocretin and hemagglutinin that are confused through a receptor that's called TCR-V4.2. So now we pretty much have, we believe we have pretty much the cause of narcolepsy. And the next question is really how the cells are being killed. And we think that this is only the CD4 interaction, so there's probably a CD8 interaction. So now we have a very nice model where the flu is involved in the trigger, and then after you have an activation inside the brain where this DQB10602 confuse this piece of flu with hypocretin, and then you develop a reaction that kills hypocretin neurons through CD8 cells. So in conclusion, we really think narcolepsy is an autoimmune disease of hypocretin neurons, and we think that there's a particular piece of H1N1 that really causes it, and then we know that there is an abnormal presentation by HLA to CD4 cells of a piece of the flu that's being confused with hypocretin. And then we know that it's this piece of hypocretin that seems to be the antigen, and that there's particular piece of the TCR that are really involved in the system. And then we think we have found this molecular mimicry between the two pieces of neurons of flu and hypocretin. These are the main people who have been involved. Is Guo Luo, who has done most of the autoimmune work and tetramer work, isolating the T-cell receptor and showing that this particular receptor, J24 and 4.2, that are clearly involved genetically and also in the recognition of the flu and hypocretin. Anna has done Holila has done most of the GWAS that shows the genetic association. Aditya, a lot of the bioinformatic analysis, and we're really looking for bioinformatic collaborators if there is anyone that's interested. Thank you so much.
Thank you very much, Emmanuel. We have about 3.5 minutes for questions.
Yeah, I'm sorry, I didn't realize there's too much always to cover. What I want to stress is that the really unique thing what we have here is that the first time that we can link the genetic of the T-cell receptor variation to the immune response to hypocretin and this piece of flu in a way that really establishes causality. So we know for sure that the abnormal response to the flu mediated through this particular piece of the T-cell receptor really must be causal because it's associated genetically.
So you mentioned that this happened actually much more often than people think, these kind of attacks on brain cells, but the orexin one's still kind of the most prominent. Are there any other, we don't seem to lose our MCH neurons or things like that? Why do you think that is?
So
why just the orexin?
Okay, there's one reason for sure, but I didn't really have time to talk about it too much. But as you saw, that it's the hypocretin that's amidated, that's the reaction. So we know now that the thymus, which weeds out all the autoimmunes, normally when you get your tolerance, you create randomly this T cell with all this recombination of receptors, and it goes through your thymus, and the thymus is used to present all pieces of your own cell, including hypocretin, and then all the T cells that react to this through your HLA and all the pieces of yourself are deleted. However, it doesn't modify the antigen, so it doesn't present the amidated part of hypocretin, which is so it's known that post-translational modifications are not presented in the thymus. So probably one of the reasons that makes hypocretin a target is that it's actually post-translationally modified in vivo. And but I'm sure that there are hundreds of other antigens in the brain that are post-translationally modified and could be targets. So I'm pretty sure that we will discover a lot of other targets in the brain that could be producing symptoms. The problem is, I think, for hypocretin, we're very lucky because it produces a clear phenotype. Same for Parkinson's disease when you don't have dopamine. But if you have no MCH neurons, maybe you're just walking around and you're kind of maybe fine. Maybe you dream a little bit more, but it's not as striking as narcolepsy, so it's difficult to recognize. So I think it's both a problem of antigen, post-translational modification, and probably also a problem of phenotype.
Thank you. One question on the back.
Thanks for the great talk. I was listening to you and thinking about the link between the orexin level and the clinical phenotypes. And you know very well that there's not a direct correlation between the level of remaining orexin in the lumbar punctions, LCR, and the clinical patterns, especially type 1 and type 2 narcoleptic. So I was wondering if you think that there would be some specific subtypes of patients who would be highly responder to the orexin agonist type and other that would deserve other type of treatments? Or do you still think that there is only one single line of patients and that maybe the susceptibility to the lack of orexin would be different between the patients?
Short answer, please.
Yes, that's a tough one. So for sure, when cases have cataplexy, we know that they have no hypocretin, and those will be responsive to hypocretin agonists for sure. Now, cases without cataplexy that have abnormal transition into REM sleep, there's definitely the two types. One, there's the same type of the type 1 narcolepsy, And there's probably a lot of people without cataplexy that have no hypocretin, but they are not as well recognized, and those will surely respond. And then there are people who have this type 2, which have other form of sleepiness without cataplexy that are probably a different cause. However, as you know, aspirin or stimulants, I mean, amphetamine works in patients even though they have dopamine. So clearly, hypocretin agonists are also going to be helpful for patients that still have hypocretin. So I think it would be helpful also for type 2 narcolepsy.
Thank you. Unfortunately, we are over time, so we have to move on to the next speaker. And